Hello, thank you for visiting my poster. I'm Vishal Oza. I'm a scientist in Dr. Brittany Lassane's lab at Department of Cell Developmental and Integrative Biology at School of Medicine at University of Alabama at Birmingham. And today I'm going to talk about mapping regulatory programs that drive glioblastoma heterogeneity in single-cell RNA-seq data. And we'll be looking at gene regulatory networks. So glioblastoma is one of the most common and aggressive form of malignant brain cancer in adults. And its management is tricky because of the heterogeneity that the patients show, as well as the heterogeneity that's present within the tumors itself. Last few years, because of the advent of single-cell RNA sequencing technologies, people have started studying glioblastoma at single-cell level, trying to dissect what drives this heterogeneity. There have been studies that have mapped this on cellular composition or the tumor microenvironment. And usually the studies look at how gene expression changes. And so either do something like differential expression or identify different subtypes and what genes are expressed by those subtypes. There is a gap where people haven't studied the relationship between genes and the transcription factors. In other words, how the genes are regulated, how that changes, which might not be directly associated with expression, but or differential expression, but the change might be driving some of this heterogeneity that we see. So in this study, we sort of integrated data from 3 different studies to see if we can integrate them or not, and then used Panda algorithm, which builds regulatory networks, and then compared the cell types that were cell type networks that were identified by Panda. So, as I mentioned before, we obtained 3 different single-cell glioma data sets, which did different studies and used different sequencing platforms. We normalized the data using the standard scan by workflow, and then we integrated the data with BBKNN algorithm. Then the clusters were identified using Leiden algorithm. And then we identified the genes that are differentially expressed in each of these clusters using Wilcoxon's rank sum test. Then once we identified those genes, we tried to look for marker genes amongst those differentially expressed genes using the cell marker database. We basically identified 12 different clusters, as you can see in figure 1. So on the left are how does the integration look across data set. As you can see, it's not completely integrated, yet it's a lot better than how they were without the integration. And on the right, you see the different cell type clusters that have been identified. So the big one on the right that you see are different kinds of astrocyte-like cells because they express certain astrocyte-like markers. The one on the left are the 2 microglial clusters, and then in the middle we have endothelial and oligodendrocyte cell clusters. And also on here you'll see the neural progenitor cell cluster. So if you see figure 2, it identifies the expression or looks at the expression of marker genes across those clusters. So this middle big red portion that you see, those are 5 astrocyte clusters, As you can see, there are certain genes that are commonly expressed across the 5 clusters, but there are certain specific marker genes. And all of these genes are usually expressed in the embryonic prefrontal cortex. So it shows the stem cell-like characteristics of the cells. Then you have the 2 microglial clusters. So there are a few genes, one cluster that shows all the high expression of all the 10 genes. One of the clusters has less number of expression of that genes. Again, these are based on these markers are expressed in sort of stem cell embryonic stem cells. So again, highlighting the embryonic nature of, or the stem cell-like nature of the glioblastoma. And then in the figure 3, what you see is the distribution of the cells that were identified from different studies. So this are different patients from one study. These 2 are from the second study, and this one is from the third study. As you can see, neither single study captures the overall heterogeneity that's present, therefore highlighting the fact that such meta-analysis are needed to be truly be able to map the heterogeneity that's exhibited in glioblastoma. So once we had these clusters, we then build Panda networks. So it requires 3 kinds of network inputs. One is the cell gene count matrix that comes from the expression data. And then we used Menta database to provide the protein-protein interaction network. And then the third network came from a Trustwor version 2 database, which basically has the transcriptional regulatory relationship. So it's a network of transcription factors and the genes. And what Panda does is it builds a network taking into account all 3 of this network information that's been provided, and it builds a final network. We then selected the significant genes that had a value of more than 2. Once we had this individual Panda networks for each cell type cluster, we then built a multinet wherein we combined all this, where each layer was a different cell type cluster. And then we looked at how the degree of the each node changed across the clusters, and then calculated the deviation. So basically, the score, a deviation score of 0 means that node degree doesn't change much, whereas the higher deviation score meant that those degrees changed a lot across the layers. So what this tells us is how does the neighborhood of that gene change. So it doesn't look at just the expression, it's the association with the other genes and how that changes between cell type. And we identified top 10 genes or transcription factors that change across layers, and some of them have been implicated in other cancers. So for example, SP3, the topmost transcription factor, has been expressed or is expressed throughout all the tissues. If you look at the human protein atlas expression data, so it's not the expression that's driving, but the genes that it regulates changes between clusters. And so this approach allows us to go beyond just the expression and look at how the relationship changes between clusters or between cell types. In future, we will be continuing to add more data set and build this network and look at how the regulatory relationships change. But one of the limitations, I would say, is not all data sets have raw data available, and therefore that limits what we can do with in terms of integrating the data. So ideally, having this raw data would allow us to do pre-processing in a similar way, and then find cell types that are common across and look at cell types that differ, even though they might be identified as similar or same type in the original studies. So we will be trying to do that and then compare different integration methods to see how that works, and expand on this heterogeneity. Is it defined by cell types, or how the regulatory programs change? Thank you for coming. If you have any questions, feel free to ask. Thank you.